Another day, another story. Buzz off, new genetic technology developed to stop malaria-spreading mosquitoes. Scientists at the University of California, San Diego have developed a mosquito suppression system, Ifegenia, which uses CRISPR technology to eliminate female Anopheles gambiae mosquitoes, the primary transmitters of malaria in Africa. The system disrupts a gene that controls the mosquito's sexual development, thereby halting the spread of the disease. The researchers are optimistic that this approach, which is safe, controllable, and scalable, can be adapted to suppress other disease-spreading species. As envisioned, this first-of-its-kind African mosquito suppression system would reduce child mortality and aid economic development. Malaria remains one of the world's deadliest diseases. Each year, malaria infections result in hundreds of thousands of deaths, with the majority of fatalities occurring in children under 5. The Centers for Disease Control and Prevention recently announced that 5 cases of mosquito-borne malaria were detected in the United States, 4 in Florida and 1 in Texas, the first reported spread in the country in 2 decades. Fortunately, scientists are developing safe technologies to stop the transmission of malaria by genetically editing mosquitoes that spread the parasite that causes the disease. Researchers at the University of California, San Diego, led by Professor Omar Akbari's laboratory, have engineered a new way to genetically suppress populations of Anopheles gambiae, the mosquitoes that primarily spread malaria in Africa and contribute to economic poverty in affected regions. The new system targets and kills females of the A. gambiae population since they bite and spread the disease. UC San Diego researchers have developed a new technology to suppress Anopheles gambiae, the mosquitoes that primarily spread malaria in Africa and contribute to economic poverty in affected regions. Published on July 5 in the journal Science Advances, first author Andrea Smidler, a postdoctoral scholar in the UC San Diego School of Biological Sciences, along with former master's students and co-first authors James Pai and Rima Apte, created a system called Ifegenia, an acronym for Inherited Female Elimination by Genetically Encoded Nucleuses to Interrupt Alleles. The technique leverages the CRISPR technology to disrupt a gene known as femaleless, FL, that controls sexual development in A. gambiae mosquitoes. Scientists at UC Berkeley and the California Institute of Technology contributed to the research effort. Ifegenia works by genetically encoding the 2 main elements of CRISPR within African mosquitoes. These include a Cas9 nuclease, the molecular scissors that make the cuts, and a guide RNA that directs the system to the target through a technique developed in these mosquitoes in Akbari's lab. They genetically modified 2 mosquito families to separately express Cas9 and the full targeting guide RNA. An artist's depiction of Ifegenia, a new technology developed at UC San Diego that uses CRISPR genetic editing to disrupt a gene that controls sexual development in the larvae of African mosquitoes. We crossed them together and in the offspring, it killed all the female mosquitoes, said Smidler. It was extraordinary. Meanwhile, A. gambiae male mosquitoes inherit Ifegenia, but the genetic edit doesn't impact their reproduction. They remain reproductively fit to mate and spread Ifegenia. Parasite spread eventually is halted since females are removed and the population reaches a reproductive dead end. Traditional methods to combat malaria spread such as bed nets and insecticides increasingly have been proven ineffective in stopping the disease's spread. This technology has the potential to be the safe, controllable, and scalable solution the world urgently needs to eliminate malaria once and for all, said Akbari, a professor in the Department of Cell and Developmental Biology. Now we need to transition our efforts to seek social acceptance, regulatory use authorizations, and funding opportunities to put this system to its ultimate test of suppressing wild malaria-transmitting mosquito populations. We are on the cusp of making a major impact in the world and won't stop until that's achieved. Reference: a confinable female lethal population suppression system in the malaria vector, Anopheles gambiae by Andrea L. Smidler, James J. Pai, Rima A. Opte, Hector M. Sanchez C., Rodrigo M. Corder, Eileen Jeffrey Gutierrez, Neha Thakre, Igor Antashchkin, John M. Marshall and Omar S. Akbari, July 5, 2023, Science Advances. Thanks for watching.